Hello everyone, this is Dr.Nikunj on behalf of VASISHTHANEET Dedicated Academy, and welcome to this beautiful session which is Saturday night with Dr.Nikunj. Saturday night biology with Dr.Nikunj, right? So yes, there are lots of comments. Okay Uni, good evening. Here, yes, start kar rahe hum. Okay, Isha. Who is this TGJ Gaming? Okay, right. So let us start our discussion. So this session is dedicated for those all aspirants who needs a proper guidance, who needs a proper content. And this session is little bit based on the standard 11th biology, right? So in this session, what we will discuss certain points which are in the part of 11th, but it is an integral part of 12th as well. Why? Because in your standard 11 you have a certain topics which affects on your 12th study, which affects on your content basically, which is very crucial for the understanding of genetics as well, right? So that is the important thing, and we have to discuss all of this aspect related with the genetics. Okay, so let's get started. So all those 11 students, please attend this lecture very carefully. If you are in the 12th, also you have to attend this lecture very carefully. Why? Because in your 12th examination in NEET as well, you will get this certain points, and here we will discuss some advanced level fundamentals, advanced level talk related with the nucleic acid. So let's get started. So are you ready everyone? Please raise your thumb. Raise your thumb everyone. I'm not getting, I think the video is delayed. The streaming is somehow delayed. Yes, I get this. Yes, so let us discuss and see what are the topics for today, right? So topics are little bit related with the nucleic acid. It is related with the DNA, right? So topics are like, yeah, we have to first discuss about the nucleotide, that what is nucleotide, what is the composition, how they works. This second topic that we have to discuss here is a nucleic acid. Another one we have to discuss is the Chargaff rule, and last but not the least, it is the types of DNA, that what kind of DNA we get, how the DNA is changed from 1 to 1, what are the AT type DNA, what are ABCDZ DNA, right? This seems something new for you people, but they are the integral part of our life, because the DNA is the life governing particle. DNA is the life governing force who governs our life as well as maintain all the metabolic activities. So we have 4 topics, and now you people get me very carefully, that we have a 4 topic and we have to stand along, right? Yes. So good evening, almost it is a night. Good night. No, good night does not mean this is a good night, right? We have to stay long for 12 o'clock, maybe before 12 o'clock we will try to finish. And if you have a doubt, we have additional doubt sessions, right? So after finishing this session, we will go for some doubt discussion, and that you will bombard all of your doubt that you have get, right? So let us start with the first. You can ask in general in the chat box, right? If you don't get with the nucleic acid or nucleotide or anything, so you can discuss that as well. So let us first start with the nucleotide. What do you mean by nucleotide? What is nucleotide? We have a DNA. DNA is a nucleic acid, and this all nucleic acids are made up of nucleotide. That means the nucleotide are monomer. Nucleotides are monomer. Monomer of what? Monomer of the nucleic acid. So if you have to learn something, you have to go for a base. If you want to learn a living one, you go into the detail and ask the basis of cell. If you want to learn about some chemical funda, you go on the monomeric basis, that what are the components, by which component this particular chemical is formed. So for that reason, we have to first go for a monomeric discussion, that we have to discuss nucleotide briefly, then and then only you can understand a nucleic acid, because they are the monomer of what? They are monomer of nucleic acid. Okay, right. What is the other discussion? Nucleotides are basically made up of 3 units. Yes, it is a monomer, but this monomer itself made up of 3 units. If you remember of your 11th class, the discussion of carbohydrate. So carbohydrates are what? Carbohydrates, just like cellulose, the starch, glycogen, all these are the polysaccharide. But each and every polysaccharide, they are made up of the monosaccharide. The monosaccharide is the prime base which is important for the construction of a whole polysaccharide. But in this discussion, what we have to discuss? We have to discuss about the nucleotide, which is not as like the monomers, like in the carbohydrates, as like you can say the monosaccharide. It is not like the amino acid. You know that amino acid is the integral part of a protein. There are different type of amino acid. We have a 20 type of amino acid inside our body, and that 20 type of amino acid will makes a clear cut protein, right, which is enzymatic, which gives us enzymatic effect as well. Clear? So protein synthesis, the protein is something different, carbohydrate is different, in which a monomer itself a unit. But in a nucleic acid, the monomer, which is a nucleotide, has 3 separate units. So let us discuss those 3 units. The first unit is nitrogen base. What are the nitrogen base? We will discuss them. But nitrogen base are basically the molecule. These are the molecules contain a nitrogen atom. That's why it is known as the nitrogen base. Another one, we have a pentose sugar. The pentose sugar is the part of carbohydrate, yes, but it will make a base of a nucleic acid. It will come into the structure of nucleotide. And last but not the least, we have a phosphate, which in general a phosphoric acid, because the phosphate itself, it is a derivative of phosphoric acid like H3PO4. When you put them into the aquatic medium, now it will be the PO4 minus 3. So I hope you all getting, right? So if you have any confusion, if you have any other doubts, you can ask right now. Yes, Kalpesh, exactly. Nucleoside plus phosphate, it is a nucleotide. Okay, very good. Yes. Okay, so the next thing we have to discuss is the nitrogen base. So nitrogen base is our first discussion. See, there are 2 types of nitrogen base. 2 types of nitrogen base. What are those 2 types? The first one is a purine. So we have a purines on one hand, and another are the pyrimidines. Okay, so we have a purines as well as the pyrimidine. Now let us discuss about the purine first, and then we will go for the pyrimidine. So in this session, first of all, we have to discuss about the basics of purine, basics of pyrimidine, and then we will discuss how you can differentiate them. So let us start. Purines are what? Purines are the 2-ring structure. It is a 2-ring nitrogen base, where the pyrimidines are 1-ring nitrogen base. Now the question is, how this purine made? How can we make this pyrimidine, and how we remember them? First of all, let's talk about the examples. That what are the examples of the purine as well as pyrimidine? So in general, we have a 5 kinds of purines as well as pyrimidines. So 5 types of nitrogen base we have: adenine, guanine, cytosine, thymine and uracil. Among them, adenine and guanine, these 2 are what? Adenine and guanine, these 2 are the purines, where the pyrimidines are cytosine, thymine and uracil. Now you get a confusion, sir, why purines are 2 and pyrimidines are 3? Because that is the discriminatory in between the DNA and RNA. We can differentiate DNA and RNA on the basis of pyrimidine only, right? We will discuss them. So first of all, let us look that why this 2-ring structure and how this 2-ring structure are formed. See, so this is the first ring. This little bit look like as the components of the benzene, like this. Okay, so we have a two ring. You can clearly see this two ring. Yes, you will ask me that, sir, this is not ring, this is the one part, this is second part, this is the third part. We make a space in between, but this space are what? This space are nothing but a place where nitrogens are accommodated. Clear? So we have a 4 nitrogen, right? So purine is the structure compose what? Purine is the structure compose 4 nitrogen atoms. Okay, it compose 4 nitrogen atoms. Where these 4 nitrogen atoms are? It is placed on the place like 1, 3, 7 and 9. These are the place acquired by the nitrogen atom. Let's put them. So this is the nitrogen here. 1 nitrogen will be here. 1 nitrogen will be here. 1 nitrogen will be here. So let's give a number. Okay, so numbering, how you can do numbering? Clear? So you have to start the number from here. Why? Because this compose the double bond as well. So you have to start numbering from active group as well as the double bonds. Clear? So here you have to start a number. So this is 1, 2, 3, 4, 5. Now question is, which one is 6? Here or here? We have to distribute a rule that you have to first finish a ring, and after finishing that ring you have to go for the next one. So if we place 5 here, now we have a 6 on this, 7 on this, 8 and 9. Look, at the first place we have a nitrogen, at the third place we have a nitrogen, at the 7th place we have a nitrogen, and instead of that we have a nitrogen at the 9th place also, and at the 9th place there is a hydrogen attached with the nitrogen. This hydrogen, the hydrogen of 9th nitrogen is very important. Clear? So this is what a fundamental basic structure of a purine. Are you getting my point? Yes? Okay. So thoda sa थोड़ुक हिंदी में. Okay. Yes, you are the student of English medium. You should know the English properly. Okay. Yes. So what we have a first, third, 7th and 9th. One more thing I want to share you, that please make sure that our voice should be cleared, right? If not, then you have to comment, right? Our live telecast should be proper. If not, then you have to comment, right? So please make sure कि सब कुछ सही से चल रहा है. Okay, I accept your request and I will now talk in the Hindi as well as English. Okay. Yes. Okay, so it is okay with the English. No problem. So this is the first ring. First ring like a purine. Now let us look on the pyrimidine. So what is pyrimidine? Pyrimidine is a one ring structure. That means it is not that broad as like the purine. That means nitrogen number should be less. Right? How many nitrogen there? The nitrogen number is 1 and 3. Right? So there are 2 nitrogen instead of 4. We'll learn and see that purines are a nitrogen base in which we have a 4 nitrogen atom. On the other hand, we have a pyrimidine which composes 2 nitrogen atom at the level of 1 and the 3. So let's do it. So this is the ring. Look, this is what we have a ring. The single ring composes a nitrogen, right? Where the nitrogens are. This is the first nitrogen. This is the second nitrogen. So if we give a number, what should be the number? This is first, second and third. One more important thing, that the characteristic of this pyrimidine is that on a second carbon we have a ketone group. Okay, so that is the identificatory key for a pyrimidine. One ring is also a characteristic. So this was a discussion very basic about the nitrogen base. Now we have 2 type of ring. Now we understand solve fully that how this ring are and how this ring made, right? But one more thing that we have to concern is that where should the component placed by which we can differentiate them, because under the composition of purine we have a 2 adenine as well as guanine, and under pyrimidine we have a 3 cytosine, thymine and uracil. So let's look. So let's quickly draw a figure. So should I say this is what? A purine ring, because two rings at the first, third, fourth and ninth, sorry, not a fourth, it is a seventh and ninth place we have a nitrogen. Now the sixth number place is very important, because at the sixth place, see, this is the sixth number place, and on the sixth place, okay, on the sixth place, what we have? On the sixth place, if we get an amino group, okay, what kind of group? Amino group, then it is known as a purine, right? If you have an amino group here, then it is what? It is a purine, but what kind of purine? It is an adenine, okay? So this is the classificatory way. If your examiner asks you a question from anywhere, you can now answer. If you have a two ring structure, if you have a 4 nitrogen, if you have an amino group at the sixth place, then you can say this is the structure of adenine, because it is very clear now. Another one is the guanine. So yes, we know about the adenine now, right? But what about the guanine? How we can assume that it is a guanine? So fundament is also very clear. See, if you have an amino group, these are the tricks by which you can understand. If you have an amino group, then it is an adenine. If you have a ketone group, right, this is the ketone. So if you have a ketone group, then it is what? This is the guanine. Okay, then it is the guanine. So this was the discussion related with the purine. You now know that how you have to make the purine. Another thing is a pyrimidine. Now pyrimidines have 3 options. Let us draw all these 3 rings. Okay, so now we have a 3 rings like this. Yes. Here is nitrogen, right? Here is also a nitrogen. So is it clear? Yes, Avni, please tell me from where I have to repeat. Okay. Right. If you want to repeat something, please message and please tell me that from where I have to repeat. Yes. Okay. So we have a 3 example and we draw 3 rings. Yes. Okay. Okay, Deep, we have to see a chat. Clear. But a little bit I focus on the study right now, because the session should not proceed more than 1 AM in the night, because that will be more, you can say, hurdleful for you. Okay. Yes. I will give note. I will give time for note. Okay, Avni. Okay. See. So we have a 3 type. We have a 3 ring. So there is 1 and 3rd place. Compose what? The nitrogen. But at a 3rd place, we have a hydrogen as well. As like the purines, we have the classification according to the place. Place number what? Place number 6. If at the 6th place, we have amino group, then that should be what? Like this. If we have an amino group, then that should be the cytosine. Okay, if we have an amino group, that is a cytosine. If we have a ketone group, then it is a uracil. Now another discussion. We will discuss that on the basis of pyrimidine, you can classify DNA, you can separate the DNA from RNA. You can identify DNA separately, RNA separately. How? The adenine, guanine, cytosine, they are common. They are common for what? They are common for DNA as well as RNA. So if you have a DNA, if you have a RNA, no matter what you have, right? If you have a nucleic acid like DNA, like RNA, then you have what? Adenine, guanine and cytosine. Compulsory. But that is not with the thymine and uracil. If you have a RNA, then you have a uracil. If you have a DNA, then you have a thymine. If you have a DNA, it does not have the uracil. If you have a RNA, it does not have the thymine. Clear? So that is the understanding. Right. So this is what the talk related with the nitrogen bases. So cytosine, thymine, uracil, these 3 are the pyrimidine, but in a one structure, there should be the cytosine If RNA, then cytosine is with uracil. If DNA, then cytosine should be with the thymine. So that is the discussion. Let's move ahead. Yes. So now we know that if we have the ketone group at the sixth place, we have a uracil. But what about thymine? Right? What about the thymine? So thymine is just like a uracil. Why? Because structure should be matched, because cytosine has to bind with the uracil as well as thymine. That means there is a structural similarity, but not that much. We have a structural similarity that on a sixth place we have a ketone group. Then a question comes, sir, how we can now differentiate them? It is very easy. See, the thymine is also called as methylated. It is a methylated uracil. What do you mean by methylated uracil? It is very clear. See, if you have a uracil, add a methane group, then it is what? Then it is a methylated uracil. So if we place a methane, where? At the fifth place. If you place a methane on a fifth place, now it is known as a thymine, because it is now a methylated uracil. Are you getting my point? Yes or no? Please tell. Yes. Okay, use yellow color on the place of green. Yes, sir. But I'm using the yellow. Okay. Right. So let's move ahead. So we have now the conclusion very clear that we have the purine as well as pyrimidine. If that is a purine, okay, if that is a purine, then we have a two-ring structure. Then we have a two-ring structure. But let's look. The structure is different. Yes, structure is same actually. This is the first, this is the third, this is seventh and ninth. Right? But the hydrogen with 9 is very important. Why? We will discuss later on. At sixth place, if we have an amino group, then it is adenine. If we have ketone instead of amino, then we have the guanine. Clear? The another one is the pyrimidine. Now structure of pyrimidine is little bit inverted. Okay, no matter how it is, but if you start first, second and third, right? So at the sixth place, if we have an amino group, then this pyrimidine is cytosine. If we have the ketone instead of amino, then it is a uracil. And with the ketone group, if we have an methyl group at the fifth carbon, then now it should be what? It is a thymine, because thymine is a methylated uracil. So now we conclude all of the nitrogen base, and now we know that which one will come into the category of the DNA's nitrogen base, like adenine, guanine, cytosine and thymine. And now we know that what kind of nitrogen base came under the RNA structure, like adenine, guanine, cytosine and uracil. So let's move ahead. But before that, I want to tell you some rules. What rules we have? Okay, spelling mistake, no problem. See, what have a rule we have? The nitrogen base always binds with what? It always binds with the pentose sugar. Where? On the pentose sugar. But where? From where? See, there is a first carbon of sugar and the ninth number nitrogen of a purine and third number nitrogen of pyrimidine. And that's why we will discuss that a nitrogen number 9 and a hydrogen of that 9 nitrogen is very important. Nitrogen number 3 is very important, and on that nitrogen the hydrogen is very important. Why? Because they are bind with the pentose sugar. How, why and on which means we will discuss them later on. So let's move ahead. What we have? This is adenine, guanine. See, if we have a amino on 6, then it is adenine, otherwise it is a guanine. So this is the component of a uracil. You can take a screenshot and look them. Okay, so let us start a discussion related with the pentose sugar. That is the second one, because as we discuss the nucleotide, see here, the nucleotide is made up of nitrogen base, pentose sugar and phosphate. So we already discussed about the nitrogen base. So our second topic here is the pentose sugar. So let's look. Yes. So what is pentose sugar? The pentose sugar is a carbohydrate. You all know about that. The pentose sugar is which kind of carbohydrate? It is a monosaccharide, right? These are the monosaccharide. Now, what is the molecular formula of monosaccharide? That is also important. So, molecular formula of an pentose sugar is CN H2O N, right? What this N? N is the representative. It is the representative of the carbon number, or you can say the number of carbon. Now, if you have the carbon number N is equal to 3, then it is a triose sugar. It exceeds up to the 7, that is the heptose sugar. But what is pentose sugar? Pentose sugar means you have a N is equal to 5. When you have a 5 carbon in a monosaccharide, then it is what? Then it is the pentose sugar. Now, as we titled it as a pentose sugar, we have to look for whole 360 degrees of this topic. But among this whole 360 degree, all are not important. Okay, there are few points are important, right? There are few sugars are important that will be the part of the DNA or whether RNA. Clear? But let's look. So, if you have N is equal to 5, then it is C5H10O5. Is there any difficulty? If you are agree, please raise your hand. Yes. So, we have C5H10O5. Clear? That is the structure of pentose sugar. Now, what should be the example? Examples are like ribose. We have a deoxyribose. We have a ribose. We have the ribulose. And also we have a xylose. Now, under this 5 pentose sugar, how many are important for us? That is ribose as well as deoxyribose. So, ribose and deoxyribose, these are the important sugar for us to form a nucleic acid. But we have to talk about whole 360 degree of pentose sugar, that we have to ensure here that you have to no need to go search for the pentose sugar or any kind of other pentose sugar, right? So let us discuss about these three. The arabinose is used for the gum arabic. Okay, found in the gum arabic, that is the material. In the successive or the next Saturday session, we will discuss them. Ribulose is what? Ribulose is the sugar found in the Rubisco. Okay. So please tell me what is Rubisco, right? That is your question, right? What is Rubisco? Where Rubisco came? What is the identity of Rubisco? So if you know, let us discuss. Rubisco is the world's most abundant enzyme. Okay. If we have 100 enzymes in the biosphere, among that 100 enzymes, 80 are nothing but the Rubisco, because Rubisco is the most abundant enzyme. Okay, so Rubisco, clear, it is the most abundant protein as well. Clear, in the biosphere, it is the most abundant plant protein. So this Rubisco, which is the photosynthetic enzyme, right? Rubisco is stand for ribulose biphosphate carboxylase oxygenase. Clear? So here see, the R is stand for ribulose. Clear? So ribulose is the sugar used in what? It is used in the formation of Rubisco. The last but not the least, that is the xylose. And xylose is found in what? It is found in the cell wall. The xylose is found in the cell wall. Is it clear to everybody? See, so this is what? The arabinose, ribulose and xylose, so on. What about these two? These two are used in RNA. Which one? Ribose. And the DNA, deoxyribose. Clear? Now, you already know the difference between RNA and DNA, because there are only two different present in RNA as well as DNA. All the DNA compose adenine, guanine, cytosine as like in the RNA, but all the DNAs compose thymine instead of uracil, and RNA compose uracil instead of thymine. So that is only difference. Are you getting my point? Right? That is only and only difference. On the other hand, what we have? On the other hand, this pentose sugar is different. In RNA, there is a ribose, and on DNA, that is the deoxyribose. So we have a two difference. First difference on the basis of nitrogen base, and second difference on the account of the pentose sugar. Right? Nitrogen base, thymine for exclusively in the DNA, uracil for the RNA. Pentose sugar ribose is for the RNA, and deoxyribose is the sugar for the DNA. Are you getting my point? Right? If you know about this fact, then okay. If you don't know, then please like this video, right? So this is the discussion, very basic about the pentose sugar. But our interest is only and only into ribose and the deoxyribose. But prior to that, let us understand one more fact, that any carbohydrate you get, right? What is the common formula of carbohydrate? That is CX H2OY. Okay, H2O 20 नहीं लिखा है. This is the H2O, right? CX H2OY. Now, as the name carbohydrate suggests, carbohydrate, it is a hydrates of carbon. So water and carbon is there. Yes, the number of carbon and number of water can be up down, but the hydrogen gem oxygen ratio, see this H2O is all Y, so hydrogen and oxygen ratio should be 2:1. That is the rule, but it is not followed by everyone, right? It is okay for other monosaccharide, other all these polysaccharide, disaccharide, oligosaccharide and on, but it is not true for one and only our deoxyribose sugar, and it is makes it special. It makes it very stable. Clear? So this deoxyribose sugar, is what? It is C5H10O4. That means you can see here hydrogen are 10, oxygen are 4. That means the ratio of hydrogen and oxygen, which is what? Which is 2:1, is not followed. So that's why it is very special, and it makes our life steady, because due to this lack of one oxygen makes the DNA molecule much stable than any other one. Okay, so let us move ahead. What about the ribose? Ribose is a C5H10O5. It is a regular sugar, regular pentose. Let's put those all carbon in a one chain. Okay, so we have a 5 carbon. Now another classification of carbohydrate that you might know, and if you don't, right, please comment in the description below. We will make a separate video for that also, right? Or we get that topic in the next Saturday evening for the biology talk, right? So let us see. We have a ribose. Ribose has a 5 carbon. These 5 carbons are put in a row. 5 carbons we have, right? Now carbohydrate, this carb should be of aldose mean, and it should be of ketose mean, right? What do you mean by aldose? Those sugar compose a aldehyde group. That is what? That is a aldose. Those sugar compose a ketone group. That is what? That is a ketose. Clear? So are both the sugar, ribose as well as deoxyribose? Yes, ribulose was the ketose. Okay. Here both the ribose as well as deoxyribose, they are what? They are the aldose sugar. If you get any doubt, please write on the other paper. At the end of this session, we will discuss all of your doubt, right? So let's put all them together. So we have C double bond O here, aldehyde group. Other are the hydroxyl group, and remaining valency of carbon is fulfilled by what? It is fulfilled by the hydrogen. Yes. Okay. So this is the linear structure. But sir, we as know the ribose sugar, it is not a linear, it is a circular, right? And there is a ring called as a furanose. How this ring is formed? Let us look. Right? Don't mug up any ring, because there is no need for mug up when you have a little understanding. Right? When you put little emphasis on any topic, you get them very clear, very crystal clear on your mind. Right? So let us look. We have oxygen on a first carbon. first, second, third, fourth, and fifth. Clear? What happened here? So this oxygen which is present on the first carbon, it share the oxygens, the first carbon share its oxygen with the fourth one, which is the second last. Now you will tell me that why second last only? Because if we share it with the last one, it will make a structure imbalance. Okay, it will cause for the imbalance of structure. So that's why on the fourth carbon and first carbon there is a ring form, see, and the OH from here now shift on the first one. Look it carefully. If you look this carefully, you will get there is a ring. See, here is the ring. Okay, so this is whole ring made up of how many contender? There are 5 members. Okay, there are 5 members. What those 5 members? See, 1 oxygen and 4 carbon. So any of the rings compose 5 member. Okay, any of the ring compose 5 member, that is known as a furanose ring. Okay, and please write down a comment inside a section box that what do you mean by a 6-member ring. If we have a 5-member ring, then it is what? Then it is a furanose ring. If we have a 6-member in whole ring, that is known as the pyrinose ring. Are you getting me? Right. So the pyrinose and the furanose, we have a 2 type of ring. Right. If you get all the fact, then please raise your hand. Yes. Okay. So we have a 5 members ring. Let's put them all together. We have oxygen here. Yes. So we have oxygen. See, oxygen is the link between first carbon and the fourth carbon. So oxygen has 2 valency. We split that 2 valency. Okay. So these 2 valencies are of oxygen, right? The 1 valency of oxygen is bind with the first carbon and 1 is with 4. So why you give number 1 to this and not this? Because always remember, the ribose, deoxyribose, both the sugar are the mean of dextrose sugar. What do you mean by dextrose sugar? All the sugar rotate in the clockwise manner, that is known as the dextrose sugar, and that's why we put them in such a manner. Okay, so oxygen's this valency is the carbon is the first one and this is the fourth. Clear? So let's finish this. So we have a 2 carbon remain under. So ring will be like this. There is no mug up here. Right? If you learn in a such a manner, you don't have to need mug up a biology. Otherwise, there are so many people in the market who says biology is very easy. Biology is the subject of a mug upness. No, it is whole about the soul understanding. If you make them of yourself, if you make them read them, if you practice them from the heart, it is all about the understanding. Okay. So this is the oxygen and we split these 2 valencies and we form a structure. That structure is of a ribose. Let's put those all here. At the first carbon, what you see? There is a OH and H. Okay. We put them. At the second, we have a H. We have a OH. So we put also them. We have a H and OH at this place also. So we put them here. And we place H and CH2OH. See, this is the CH2OH, whole 5th carbon is remain outside the ring. So that will be placed at that particular point. Now the question is, sir, this is a ribose. What do you mean by deoxyribose? See, as name suggests, it is a deoxyribose. So the structure is very similar. Okay, linear structure as well as cyclic structure. They are very similar with what? With your ribose sugar. So let's first draw the ribose here and then we will make a change. Okay, so this is nothing but a ribose sugar. Okay, this is the ribose sugar. Yes. Now what we have to look for? See, this is all ribose. As name suggests, it is a deoxyribose. Okay. That means we have to remove oxygen, right? But from where? From the second carbon. Okay. If you remove the 1 oxygen from second carbon, that means now there is no hydroxyl group here. Now there is no hydroxyl group here. Instead of hydroxyl, we have a hydrogen only, right? So one reactive group is now left, and whenever there is a left turn of, you can say, whenever there is a leaving of any reactive group, component will become more and more steady, more and more stable. Clear? So this is the discussion related with deoxyribose sugar. So what is deoxyribose sugar? It composes less oxygen than a ribose, and that is what, from a second carbon. So carbon number second is very important. When you erase one oxygen, when you decrease down one oxygen from a second carbon, or in other means, if you put a hydrogen group instead of hydroxyl group, then it is what? Then it is your deoxyribose sugar. So this was a discussion related with the pentose sugar. Let us look. So here you can see, if there is a OH, then it is a ribose. If there is only H, then it is deoxyribose. As we discussed, the ribose is used in the structure of RNA. The deoxyribose, which is the another sugar used in the DNA. So these are the two discriminatory points. These are the two differentiated points on behalf of which we can differentiate DNA from RNA and RNA from the DNA. Clear, everyone? Yes. If you clear with this, please raise your hand. Please thumbs up. Yes. Okay. So let's look further. So phosphate. Right. Now phosphate is the very easy thing, right? It is not lengthier like nitrogen base as we learn about the pentose sugar. It is very easy. See, phosphate is nothing but the H3PO4. Now phosphate are basically PO4 minus 3. So chemistry teacher tell us that it is a PO4 minus 3. How you can say it is a H3PO4? It is a phosphoric acid. We agreed. No doubt it is a phosphoric acid. But wherever we have to form a bond, we have to conclude that minus, okay, negative ion. If we place a hydrogen, then that negativity is placed with the positivity. And now it is easier for us to distribute the sequence of this structure, right? So it is what is it? It is a H3PO4, that is a phosphoric acid. Clear? So it is like this: phosphate with 3 hydroxyl group, and as phosphate has the oxidizing number 5, we have a double bond oxygen. So this is all structure as we discuss here, right? So these are the 3 prime component, very useful, very essential for the formation of one and only substance, that is a nucleotide. So as we learn all the units, let's further move on the discussion of structure. So before that, we have to understand a nucleoside. Sir, our topic was a nucleotide. Yes, our topic was nucleotide, but the nucleoside is an integral part of the nucleotide as well. See, what we have? We have a nitrogen base. What we learned in the previous slides? That nitrogen base has a tendency. Tendency of what kind? Tendency to bind with the pentose sugar, not with the other one. Right? So it has a tendency to bind with the pentose sugar. So we have a pentose sugar as well here. And those pentose sugar has a tendency to what? Tendency to bind with the phosphate as well. So we have a phosphate. But the first thing first, long story short, that nitrogen base first bind with the pentose sugar, and afterward it is bind with the phosphate. So this fundamental structure form first, and then later on that will bind with the phosphate. And this union of nitrogen base and a pentose sugar are known as a nucleoside. What it is known? It is known as the nucleoside. When a nucleoside bind with the phosphate, then it is known as the nucleotide. That was our topic. So first of all, we have to form a nucleoside, and on behalf of that, under that, when we manipulate them or we manipulate the nucleoside, then and then only we can form or construct the nucleotide. As the Kalpesh was tell us that it is what? It is a nucleoside plus a phosphate. That is absolutely right. Yes. So let us move ahead, and first of all, let us understand and examine the nucleoside. As we discussed, the pentose sugar is the main thing. Place in the center. See, so this is what? This is our pentose sugar. It should be the ribose. It should be the deoxyribose. We have OH here, we have H here. So we form here a ribose sugar. Okay. So we have a form a ribose sugar. See, OH is there and the first carbon is this. As we discussed in the rule that nitrogen base always bind with the pentose sugar at the first place, at the first carbon. What happened here? The purine bind with the pentose sugar on the first carbon with the help of 9 number nitrogen. That was also a talk we made previously. So if we invert the structure look like this. If we take adenine, then here should be the amino group. Okay, if you don't get, please watch the previous few minutes, then you get that if at the 6th place we have an amino group, then it is adenine. If we have a ketone group, then it is a guanine. Okay, so we have this two. What happened here? The attraction in between what? Attraction in between the OH and H. That result what? That result us as a formation of bond, and that bond is known as N-glycosidic bond. Okay, that is the N-glycosidic bond, and your nucleoside is ready now. Because now we made what? Now we put the pentose sugar with adenine. What should be the name? Okay, so let's write it. So ribose plus what? Ribose plus adenine. That is known as adenosine. What about if we put here a guanine? Then it will be ribose plus guanine, and that is the guanosine. Okay. So just let's place this whole nucleotide in a one nutshell and finish them. Okay, so as we learn that a nucleotide is what? Nucleotide is nothing. Very good. Yes. So nucleotide is nothing but a nucleoside plus phosphate. You getting my point? It is a nucleoside plus phosphate. So we have to put a phosphate. Another rule that phosphate, right? Remember this. The phosphate should be bind with the pentose sugar on carbon number 5. Carbon number 5 of what? Carbon number 5 of the ribose sugar. So let us discuss. If we split this one, this CH2OH, okay, you can easily write them as what? As carbon with 2 hydrogen, and here is OH. Okay, right? No problem in that because it is a CH2OH itself. So we just split them. Okay, now what we have? We have and phosphate. What is phosphate? It is a PO4 minus or H3PO4 as the phosphoric acid. See, there are 2 OH are attract now. Which 2 OH? This is the first OH. Okay, and this is the second. So OH of pentose sugar and OH of the phosphate, they attracted. And when there is an attraction in between 2 OH, how many molecules you have? You have a 2 hydrogen, you have a 2 oxygen. So that means H2O will be left from this component. Clear? So now we have a liberation of what? We have a liberation of H2O. So H2O is liberated from this place, and instead of that, what happened? It result into what? It result into an ester bond. Another bond we get here. One, we get an N-glycosidic bond at the formation of nucleoside. See, this one is the nucleoside. This is what? This is a nucleoside. As we discussed, nucleoside plus phosphate. It is what now? It is now a nucleotide. So this is our topic. We end the first topic here, but I want to more emphasize that. So you can clear any of the question from the NEET level as well as the previous AIPMT level. So this is what? The phosphoester bond, because it is the ester bond with the phosphate, so it is known as phosphoester bond. But the component now made is known as what? Adenosine with phosphate. So that is the phosphoric acid, adelenic acid. Adelenic acid, or you can write it as a adenosine monophosphate. Yes or no? Because one phosphate is there, so it should be the monophosphate. So if it is a adenosine monophosphate, then it is AMP. Okay, it is what? It is a AMP. AMP stands for adenosine monophosphate. Clear? But the adenosine monophosphate, it is the name which is very familiar. We already know this name. We already discussed the name like this, adenosine, blah, 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 for a several times. So we have to conclude them as well. What happens if I put another phosphate at this place, like this? Okay. There is attraction in between these two OH. So that will lead what? That will lead another H2O molecule liberation. If there is another oxygen molecule liberate from here, another ester bond form, right? And it result into what? Adenosine diphosphate. See, because we have adenosine. Okay, now the picture is little bit clear that for what we are going. Clear, adenosine is here, and with adenosine we place two phosphate molecules, right? So it is what? It is adenosine diphosphate, is ADP. Now the term, I think you all guess, right? Now the term for ATP, which is the energy formatting substance. Okay, so this is some extra. This is not mentioned in your topic for today, but I teach you for your betterment. So please don't go without like this video. Okay, chalo. So we have another phosphate here, the third phosphate. Oh my god, what happened here? Yes, I think the range is now over. Okay, see what happened? This 2OH also bind with the another ester bond, and H2O is liberated. So now we have a 3 phosphate, 1, 2 and 3. We have a 3 phosphate with 1 adenosine. Okay, we have a 3 phosphate with 1 adenosine, then it is known as the ATP, and this ATP is nothing but our currency for energy. Our body, as we use money for a currency to buy something, sell something, okay, as this substance ATP is our currency for a biological mechanism. Every process is done with the help of ATP only, right? The energy of ATP is very important. So I think it is very clear. Right? So if you have any doubt, then please tell me in the comment section. So here we finish the one whole phase of our discussion. Okay, one whole phase of our discussion. So, but we have to more emphasize them. If there is a ATP, then should be GTP, then should be CTP, then should be UTP. Then we don't get them. Very easy. See, if you have a ribose, if you have a adenine, then the substance is known as adenosine. Okay, and if we place one phosphate here, this is what a nucleoside it is. When you add a phosphate, that is a nucleotide, and now it is known as adelenic acid or AMP, then ADP, then ATP. So same way, if we put a guanine instead of adenine, then what it is? The guanosine or guanosine. Okay, plus phosphate. That is now what? The guanylinic acid. Here it should be the guanylinic. Okay, because it is a the nucleic acid. Okay, so instead of all the things, we also learn about the nucleic acid. So that is the second topic of us. So we are in between of a first topic as well as a second topic. Clear? So this is the discussion. Now another one is what? The ribose. The same way, if we have a ribose plus cytosine, then it is what? It is the cytosine, not cytosine, right? Not a cytosine, because cytosine is same name. Clear? And as it is a pyrimidine, we have to change the suffix. As that was a purine, we use adenosine, the suffix "ine." Okay, purine or adenosine. So suffix should be common. Clear? So in the case of pyrimidine, what we have used? We have to use idine. So cytidine, first one. Cytidine with phosphate is known as what? Cytidilic acid or CMP. What about the uracil? If we have a uracil, right, like this, it is a uridine now, phosphate, uridylic acid. If we have the thymine. Now this is very interesting. I want to know what you people gives the answer. What about the ribose plus thymine? Okay. My screen is little bit behind than whatever you write, whatever I see, right? So please, we have to wait for 10 or 15 seconds. So until that, please give the answer. Okay, what do you mean? What do you mean by the ribose plus thymine? Yes. Okay, Satya, very good. Yes. Anyone? Exactly, Meet. Yes, exactly. It is not possible. Okay. You give a right answer that it is not possible. Why it is not possible? When we use a ribose, we ensure that this molecule is ribose or a RNA, not a DNA. Clear? And thymine is a part of DNA, not RNA. That means this component should not be formed. For that, we have to use a deoxyribose. Okay, we have to use a deoxyribose with what? With thymine. Then what happened? Thymidinic acid, thymidine is formed, and when we add a phosphate under that, it is now deoxythymidilic acid. Then please try. Use a deoxyribose and place adenine, guanine and cytosine, not a uracil, because uracil also not bind with the deoxyribose, because deoxyribose is the fundamental part of your DNA molecule, does not bind with the uracil, because uracil is not present in DNA at all. Clear? So please try it. Yes, honey, it is very right. Yes. Yes, Ayushi, ribose plus thymine is not formed. Okay. Okay. So here we finish our discussion related with the nucleotide. So this is ultimately your structure of nucleotide, right? What is nucleotide? It is a pentose sugar plus a nitrogen base, and ultimately it is the phosphate group. Clear? This component, the pentose sugar plus nitrogen base, it makes what? A nucleoside, and when nucleoside bind with the phosphate, then it is the nucleotide. Okay, so whatever your review, please, if you make a note at the time, please watch, please analyze that note. I will give you 1 minute before starting the next topic, that is a nucleic acid, right? Please revise them, because we already finished our lengthier portion. Now there is a simple 3 topics which are not lengthier that much. Okay? Yes. You have a 1 or 2 minute. Okay, Tirth, you have everything mixed. There should not be a mixture. You have to make a full conscious mind and your mentality as well. Okay. Right. Those who have a little difficulty with the language, they have to cope up. So for that, you can see this video tomorrow as well, because it is a YouTube recording. You can watch anytime. So if you have, yes, we will do a short revision of full content, whatever we learn on the later on. But first of all, we have to finish them. So that's why I say that we have to discuss all of this, all of this doubt in the last minutes. Okay. So let us start with our discussion related with a nucleic acid. What do you mean by nucleic acid? How it is founded, how it is discovered. So history is the remaining part, otherwise you already discussed about the nucleic acid, right? So what is the history? Nucleic acid, as like the carbohydrate, as like the protein, it was not priorly known component. It is a new component. It is freshly formed, freshly derived component, right? So as per the history, the nucleic acid was discovered. Pause the video and gives answer. Right? Who discovered the nucleic acid? The nucleic acid was discovered by a scientist, Friedrich Miescher. Okay, so Friedrich Miescher is a scientist who discovered the nucleic acid. But where? From by the investigation, by an investigation done on the pus cell, and this is the investigation on pus cell and on the RBCs of fish. So under the discussion as well as the investigation on the pus cell, what is pus cell? It is the dead WBCs, right? So the dead WBCs, which is the pus cell, right? That pus cells are nothing but the dead WBC, and this dead WBC compose some acidic elements, some acidic component. Okay. So what are those acidic components? That acidic component later on gives a name, and that name was what? That name was the nuclein. Okay. So it is not the nucleic acid as we now know today. It was a nuclein at the time of discovery. Okay. So it was a nuclein at the time of discovery, discovered by the Friedrich Miescher in the year. You have to also remember the year, right? The year was 1869. In the 1869, nuclein was discovered. But afterward, later on, by the Altman, name was changed, and nuclein is now known as the nucleic acid. Okay, right. So same discovery for the DNA as well, because this nuclein, which is the acidic in nature, was nothing but a DNA. So discovery of DNA is also by the Friedrich Miescher. So let's put this in the nutshell, and I will revise this whole because nucleic acid now finished, because you learn about the nucleic acid, because adenylenic acid, guanylenic acid and all. So what is a nucleic acid? What is the background of nucleic acid discovery? It was discovered in the year 1869 by a Friedrich Miescher during the investigation of pus cell examination, RBCs of fish, right? And after all, they conclude a material, and that material is nothing but the nucleic acid. Name was not a nucleic acid. He gives a name which is the nuclein, and afterward Altman changed the name, and Altman gives name nucleic acid. So if examiner asks you a question that who discovered nucleic acid, then your answer should be Friedrich Miescher. But at the same time, if examiner asks you a question that who gives a name nucleic acid, so that is the answer Altman. Okay, so this is the finishing point of a second topic as well. Clear? We have a 4 topic, we finished 2 topic among them, right? So please just stay with us, right? We have you have a 1 minute to see this. Okay, and if you get any query, then you can see. Yes. Okay. So I think you all get this. Yes, Bharat Mata ki jai. Okay. Yes, there is the 12 o'clock. There is still time. 53 minutes remain in 12 o'clock. Then you have an independent day, right? And you will also get independent from my session as well. Okay. See, so the next topic is the DNA. Now, DNA is the third topic that we have to discuss. Okay, so DNA is what? DNA is a deoxyribonucleic acid. It is a deoxyribonucleic acid, right? We already discussed that what are the composition. Now we know the discovery. Discovery of what? Discovery of the DNA. DNA's discovery was done by the Friedrich Miescher in the year 1869, as we discussed in the nucleic acid, because that nucleic acid was not but the DNA itself. Another one. Who gives a name? Because the Friedrich Miescher gives name it as the nuclein. Afterward, we find that not only one acidic molecule, that is the DNA, but another acidic molecule, which is the RNA, is also part of the nucleic acid. So Altman gives a name a nucleic acid. So who gives this name a DNA? So the name DNA was given by the Jacques. Okay. We have to just discuss and the detailed history of this DNA structure. So DNA structure was given by so many times by so many scientists. The first valuable work is done in the 1940s, the era of 1940s, by two scientists. One is a Franklin and another is a Wilkins. So Franklin and Wilkins, these two scientists gives us a model of a DNA. That model of DNA was a ladder-like structure. This ladder-like structure is what? It is a DNA. Okay, look like this, right? Yes, you don't see a figure nowadays like this, but that was the most important discovery by the Franklin and Wilkins. It was seen in the X-ray chromatography. X-ray chromatography. Okay. So under X-ray chromatography, under film of X-ray chromatography, the ladder-like structure was represented by the Franklin and Wilkins. This was not right. Why? Because what they do? They freeze the DNA molecule, so now it is not rotating. They pass a X-ray light, and from this X-ray light, they get this structure. So it is arbitrary. No make a sense about the nature of DNA, because DNA is a rotating structure and it not allow DNA to rotate. That's why this was a failure. But after that, right, Franklin's and Wilkins' work was not that much arbitrary. It was too much valuable. Why it is a valuable? Because in the year 1953, two scientists, the great scientists W. Watson and Francis Crick. W. Watson and Francis Crick. They gives us a double helical model. Double helical model of a DNA. What this double helical model of DNA? This double helical model of DNA is like this. And this double helical model of DNA was proportionate on the basis of what? On the basis of the X-ray chromatographic study given by the Franklin and Wilkins. So that is also important. If Franklin and Wilkins does not give this X-ray chromatography, maybe double helical model was not introduced. Clear? So Franklin and Wilkins also have a so great importance. Clear? So this is known as a double helical model. Now, what are the properties of a double helical model? The double helical model composes two anti-parallel strands. What do you mean by this two anti-parallel strands? That means if one channel having a 5' end, another one will have 3', right? If one is like this, another will like invert, right? No two chains are similar, right? One is the stand, another one is invert. Sir, how this is possible? You can only learn this by the help of a structure. So let us do a structure. Okay, if we have a strand like this, if we have adenine, guanine, cytosine here, so that means adenine also bind with the thymine. Okay, always remember there are some rules. Rules for what? Rules for a DNA structure. That adenine always bind with the thymine with 2 hydrogen bond, and guanine also bind with the cytosine by 3 hydrogen bonds. So these are what? These are the hydrogen bonds. Okay. So these are the hydrogen bonds. So there is 2 hydrogen in between adenine and thymine, and there are 3 hydrogen bond in between guanine and cytosine. So let's look further. So here is cytosine and here is guanine. But we don't get this. Let us make a figure, and by that figurization you will definitely get the structure. So if we have a structure like this, we already discussed that how an adenine bind with the first carbon. We have a hydrogen, one hydrogen here, we have a hydrogen OH here. We already discussed that how an phosphate bind with this, right, with the bond known as the phosphoresctor bond. So this is the adenine. Another one, what we have, another one in this chain, the first chain, adenine, then guanine. Adenine follows by the guanine. So here should be the guanine, here should be the phosphate. Okay, so this is the phosphate with the phosphoresctor bond. Here should be the OH, here H and H. Clear? The third and the last one. See what we have. We have here, as we write there, it is a cytosine. Okay, it is the cytosine, and we have a phosphate here as well. And here is H and OH. Here is H and H. What happened now? The first thing is, these are the separate molecules of a pentose sugar, or you can say these are the separate molecules of a nucleotide. Okay. Then another one is, you can see they are the separate, they are not bind, but here there is a binding. See, in between adenine, this is the whole chain, here is adenine, here is guanine. So adenine and guanine are bound with something. What is this boundary? Let us discuss. So there should be a bond. What happened? See, phosphate is like this. Can we draw a phosphate like this? Yes or no? Yes. Right. So this is the phosphate. We get 2 OH in front of them, front of each other. So this 2 OH get attracted, H2O liberate here. Okay, H2O liberate here, right? And this will result into the formation of another phosphoester bond. So now these are the middle phosphate who have the phosphodiester bond. Why? Because see, the first ester bond here, second here. As here the first ester bond here, second here. So we have a phosphate molecule, which is the binder of whole the chain of pentose sugar or a nucleotide. They compose what? They compose the diphosphate bond. Okay. They compose what? They compose a diphosphate bond, by which means we have a linear structure. See, now all these three, adenine, guanine and cytosine, they are bind together. Okay. So that should be one important question. As the Franklin and Wilkins says, as the Watson says, that there is the ladder like structure like this. So under that ladder, if we straight them as like a Franklin and Wilkins, there should be a steps and there should be a backbone of ladder on which this whole ladder or the whole steps are situated. Are you getting me? So we have a ladder. Okay, we have a ladder with the step with the backbone, and you learn about the backbone. Why? So let's look here. This whole component, this whole part is like a ladder on which adenine is situated, guanine is situated and cytosine is situated. So this whole line, which is the backbone, is made up of what? Now you can say the backbone is made up of phosphate and pentose sugar, because we prove it, right? So this whole structure, in the ladder structure of DNA, we have two things. We have a backbone, we have a stair or a steps in between them. So these two backbones are made up of pentose sugar and phosphate. But we still wait to learn how these two ladders join. how these two backbones are joined with the steps. So let's learn them. Yes. So on the another side, we have what? We have the parallel. As we use a law, right? This law is what? Always adenine binds with the thymine, guanine binds with the cytosine. There is no option, no another option. We have a adenine with thymine, we have a guanine with cytosine with the hydrogen bonds. That is fixed formula. That means if here adenine, there should be thymine on the other pole, right? But what happened if we draw this thymine or thymidyllic acid, deoxythymidyllic acid like this? So now the adenine is in this side and thymine is now pushed outside. So how adenine and thymine will join? There is no possibility. For that we have to take this thymine towards inside. How you can get this? How you can make this thymine, which is now at the outside, where the bonding between adenine and thymine is not happened. Why? Because adenine and thymine are buried by the pentose sugar. There is a pentose sugar in between. You cannot bind them. If you have to bind them, you have to take those two molecules come together. So how we can do this? It is very easy. Let's make the component invertly. So we have to make an invert structure of this whole component, and your work will be now very easy. See, if we draw a pentose sugar invertly like that, if this is the first carbon, we have to make sure that rotation should be in the clockwise direction. So first carbon as the clockwise direction after oxygen is this, and on the first carbon there is a thymine. Like this. This is the first, sorry, this is the third one. Here is the fifth carbon on which by the means of phosphoresctor bond there is the phosphate bind. Let us draw all these three. First let's do all these three phosphate molecules and all. The pentose sugar as well. Yes. So here should be what? Guanine. So on the behalf of guanine, there should be cytosine. That is the law, and we have to follow them. And here should be what? Here should be, if cytosine, then there should be the guanine. So this is the base. Okay. Let us join these three sugar, or let us join these three nucleotide with themselves like this. So what happened? Here is OH. They bind with them. So what happened? Another phosphoresctor bond will form. We already discussed about them in this sequence. Okay. So this is whole mechanism. This is whole mechanism. Right. So this is what? This is the three other nucleotide bind. So this is another chain, as the thymine, as cytosine is there, as guanine is there. Okay. So this is very clear. Right. This is very easy right now. Now we have a discussion about the thymine and adenine. They bind with the 2 hydrogen bond. Here they bind with the 3 hydrogen bond, guanine and cytosine. Cytosine, guanine, they bind with the 3 hydrogen bond. See, now we have what? Now we have a 2 ladder. Okay, we have a 2 backbone. Under backbone we have a steps. Clear? So now the fundament is very clear. What is clear? That backbones are made up of the pentose sugar and phosphate, whereas these steps are made up of a hydrogen bond. Okay, that is what? That is made up of a hydrogen bond. So I think it is very crystal clear to all of you, right? Please answer in the comment section if you learn. Okay, if you learn, if you get hold the idea, please leave it in the comment section. Otherwise we will discuss each and every point, whether you not get or get, in the last few minutes after finishing the session. Okay. So we now know about the backbone as well as the step. So this is the structure of DNA. But our question is still that they are the anti-parallel. If one is a 5 prime, another is 3 prime, and the end of this 5 prime should be what? That should be the 3 prime, and the opposite one is a 5 prime. So one straight, one invert, one straight, one invert. But how it is possible? What is the criteria? What are the characteristics? Let's look how we can give this 5 prime, 3 prime names. We have a phosphate here. Okay. See, remain. Now imagine you put thousands, millions, trillions of the nucleotide together and form a chain. Okay, what happened? No mean how many nucleotide you join. The first nucleotide always remain one phosphate active group as unattached, because all those phosphate are bind with the two ester bond. There is only one ester bond with the first one, okay, with the first nucleotide. That means here is the OH which has a tendency to bind with someone. One ester bond capacity is there. Okay, there is one ester bond capacity. Now the question is, if we have one ester bond capacity, that means it is a reactive end. Okay, and reactivity is placed on what? Carbon number 5, because the phosphate itself bind with the carbon number 5. So that's why it is known as a 5' end. Okay, that's why it is known as what? It is known as a 5' end. And at the end, whether you bind the 1,000, whether you bind a billions of nucleotide, the last one, the last molecule of nucleotide, they always remain with what? They always remain with the OH, okay, on the third carbon. So that's why it is what? It is the 3' end. This is 5', this is the 3'. Let's see. These are the invert. That means here the 3', here the 5'. So I think now you get everything very clear that in the structure of DNA, strands are anti-parallel. One is 5' to 3', another one is 3' to 5'. Are you getting this point? Yes? Okay. So this is what the anti-parallel structure. Another one, we have a hydrogen bond, which is a state. But how many hydrogen bond we have? Another one is what? This one adenine bind with the thymine, this whole component with both a nucleotide known as a base pair. Okay, it is a pair of a base, so that is a base pair. Now, if you see, if this is the one strand, if this is the second one, they rotate like this. Okay, they rotate like this. They never ever cross. Here you can see there are a crossing, but they are not cross, they are rotate. Right? So that means once they rotate, they make a 360-degree angle. Clear? So what about this 360-degree angle? That is this. But you can say that they cross for 2 times when you make it like a 360-degree. No, not like that. See, if this is the 2 strand of a DNA, you can see this one. You cannot see this one. If you can, then also you cannot see this. Right? So if a DNA rotate like this, at the first crossing, it is only the 180-degree, because they first like that and now they are like that. So position is only changed. They are not a 360-degree complete. They 360-degree complete at the second crossing. Clear? So this hole is known as a 360-degree. And under that 360-degree, they need what? For a 360-degree, they need 10 base pair. And this one is known as a pitch. This 10 base pair area is known as what? That is known as a pitch. Right? So what do you mean by pitch? Pitch is the 10 base pair, which cover the 360-degree. Now what is the length? Because we learn about the structure of DNA. So what should be the length? So this 10 base pair need 34 angstrom. That is what? That is 3.4 nanometer. Clear? If 10 base pair acquire the 34 angstrom space, then what should be the space in between these two? The space in between these two, or you can say a space between 2 base pair. Clear? There are 10, so 1, 2, 3, 4. So 10 space are there, and that is the 3.4 angstrom, or you can say it is a 0.34 nanometer. So this is what the idea of the DNA structure. Now you can say, sir, you draw wrongly. No, I'm not drawing it wrongly. Why? Because as per the study, in the DNA there are 2 grooves. Okay, there are 2 grooves in DNA. What these 2 groove? One is a minor groove and another one is a major groove. See, there is one big depression that is known as a major groove. I write in the capital, so that is a major groove. then there is followed by the minor group. So let's write them into the small. So that is a minor group. It followed by the major group, also followed by the minor group. So minor follow major, major follow minor. So that is the sequence. That means the pitch is what? Pitch is the subsequent distance in between two major group or a minor group. So that is major and minor group. Maybe it is new for you, but it is important. It may be asked in the question, in the examination. So we already learned everything about this, but what about the diameter? Right? So the diameter of a DNA is 20 angstrom, or you can say 2 nanometer. Okay, it is a 2 nanometer. So let's look ahead. So we already discussed about the structure. Okay, this is the structure of DNA. Okay, this is the structure of DNA. The space in between two base pair is what? It is a 3.4 angstrom or 0.34 nanometer. The diameter should be 20 angstrom. So this is the idea. Adenine always bind with thymine with means of 2 hydrogen bond. Guanine always bind with the cytosine with 3 hydrogen bond. There is no uracil because it is a DNA. So here we finish with our topic, and that is a DNA. Okay, are you get all those points? If yes, then raise your hand. Okay, 32 minutes has to go in, or no, now it is 31 minutes. Yes. Okay, so let's see the Chargaff rule, another idea, another component, another aspect of our study. That is a Chargaff's rule. What is Chargaff's rule? First of all, it is given by the Irving Chargaff. Irving Chargaff is a scientist who gives us a law. That is Irving Chargaff's law. What this Chargaff law says? We already know that adenine has a tendency to bind with the thymine. Guanine has a tendency to bind with the cytosine by 2 bonds, by 3 bonds, all right. But can you see one difference? That everywhere purine bind with the pyrimidine. Here is also purine bind with the pyrimidine. So law is what? That everly purine should be. Okay, so purine should be bind with the pyrimidine, and every time this is applied for what? The rule is only apply for the dsDNA, double stranded DNA. If there is a 2 strain like this, if there is a 2 strain, so if at the one strain there is a purine, another one should be pyrimidine. So pure as like the purine, pyrimidine is there. So number of purine should be equivalent with the number of pyrimidine. Clear? So purine should be equivalent. The number of purine is equivalent with the number of pyrimidine. Are you getting this point? Yes. So let's look. If we make an equation, purine is equal to what? Number of purine is equal to number of pyrimidines. Okay, number of purine is equal to number of pyrimidine. What are the purines? See, adenine is equal to thymine. Let's put them. Plus guanine is equal to cytosine. So this should be the formula. Yes or no? Anyone has a doubt? If you not, then please raise your thumb. Right? Number of purine. Adenine is equal to thymine. So you can erase them. Guanine is equal to cytosine. So addition of purine is equal to addition of pyrimidine because they are joined together, and it is a unanimous rule. Clear? So if you get this, please raise your thumb. Is there any difficulty? Yes, Kalpesh. Yes, here. You are more advanced right now. Here. Okay, Satya, what is minor group? What is major group? We will discuss them in the later part of this session in the last. Yes. So if this is right, then A plus G by C plus T or T plus C, as we place them below, then it should be equal to what? Right, you can move them below this A plus G, and you can move A plus G below the T plus C. So T plus C by A plus G, all should be equivalent with 1. Okay, so all should be equivalent with 1. Right, so this is what? This is the Chargaff's rule. Okay, this is the Chargaff rule. But where this Chargaff rule is useful? So I give you a question, I give some minutes, and you have to give me answer. Okay. So this is the practice question. Let's do. If I say the guanine is equal to 27%, then what about what should be the percentage of thymine? Yes. If a guanine is equal to 27%, let's change the question. Then what about the adenine? Right? So please try by yourself and do this question. It is very easy. Okay, and place that in the comment section. Okay, Meet. Thank you, Deep. I just need a focus, not comments. Okay, Satya, it is 27%. Kalpesh 23, Teet 23, Jamin Mistry 23, Heer Patel 23. So majority are with the 23. What about the other ones? Where they are? Is they are sleeping? Because it's now 11 and 34. You don't have to sleep right now. Okay. Please invest your concentration. That will invest you for whole life. Yes, so majority gives the answer 23. Is this right or not? Let us discuss. So, as we discuss here, the fundamental idea is what? That adenine plus thymine is equal to what? Guanine plus cytosine. As now we know, now what we have to discuss? We have to discuss about the adenine. This is the wrong. Adenine plus guanine is equal to thymine plus cytosine. If we have a adenine, guanine percentage, we should get with the adenine. See, if those two are equivalent, adenine plus guanine is equal to thymine plus cytosine, that means they both are a 50-50. 50% molecule is of adenine plus guanine and 50% of thymine plus cytosine. Then it is 50 plus 50, 100%. Right? So place this. So can I say adenine plus guanine is 50%? Right? Adenine plus guanine, which is 27%, is 50%. So adenine is equal to what? 50 minus 27. So answer should be 23%. So this kind of question they can ask, but you should prepare with your sole understanding that you can crack this question. Okay. So yes, it is very good. The answer is very right. So let's move ahead and discuss something. What about the ratio? If we say, yes, if we see that adenine plus guanine by thymine plus cytosine is 1. But what about the invert? Adenine plus thymine by cytosine plus guanine. Obviously, this is not 1 because these two are not with this two. Adenine is like thymine, so you can say yes, adenine and thymine, they are similar. But is adenine is similar with cytosine? Is thymine with similar with guanine? No. That means the ratio should not 1. Okay, and it will gives us and base on which we can divide we can give a types of DNA, and that is our next topic. Okay, so last but not the least, it is a type of DNA. How you can give a type of DNA? On the two bases, right? So the number one is on the basis of number of the bases like AT and GC type DNA. So first of all, let us discuss about the AT type DNA, okay, which is present in the eukaryotic, right? It is very simple. See, if we get this particular equation, it is not equal to 1, but it is specific, or you can say it is a species specific. It is specific for every species. If you take a human, if you take a bacteria, if you take anything, it is specific for the each and every species. Clear? If we are a human, we have a separate one individual specific number for it. Bacteria compose other. In humans, what it is more than one. Okay, for a human, for the humans, A plus T by G plus C is equal to 1.52. Can I just simply say it is a greater than 1? Okay. So if that is a greater than 1, that indicate what? In our body, adenine and thymine are more than a guanine and cytosine. So if adenine plus thymine means these two are more than guanine and cytosine, that kind of DNA is known as the AT type DNA. One example is a human being, and that is what? 1.52. Are you getting this? Yes. Okay. So this is what the idea all about. So let's erase them and go for the second. Let us go for the second. That is a GC type DNA. What do you mean by GC type? First of all, it is found in the prokaryote. The first one is AT type DNA, which is found in the eukaryote. The latter one, GC, is found in the prokaryotic. So prokaryotic DNAs are a GC type. What do you mean by this? If adenine and guanine are lesser than adenine and thymine is lesser than guanine plus cytosine, means the lower number are more, and then when the lower is more, then value should be less than 1. Okay, like in E. coli. In E. coli bacteria, the value is 0.92. That indicate that the guanine and cytosine percentage is much more than the adenine and thymine. Clear? So that is a GC type DNA. It should be new for you, but it is important for the understanding, sole understanding of genetics, because without this you cannot understand a genetic at all. Okay, so this is the idea about the first base. The another basis is what? On the basics of the structure. How the structure is there? What is the number of base pair? What should be the number of pitch length? As we discussed, the pitch length of our DNA is the 34. But human DNA, see, the human DNA is what? Human DNA is the B type DNA. What it is? The B type DNA, right? So we will now discuss about the types of DNA. So AT and GC we already discussed. On the basis of structure, there is A type DNA, B type DNA, C type DNA, Z type DNA, and the D as well. Clear? So let us write what are the bases. On the base of pitch, okay, under which we have a 34 angstroms pitch. We already discussed that one pitch of R is composed how many base pair? So number of base pair is 10, right? We already discussed in this particular session that one pitch means when a DNA molecule rotates for a 360 degree, or it makes a one rotation, that compose 10 base pair, known as a one pitch, right? So this one pitch is lengthen about 34 angstrom and it is composed 10 base pair. Yes. Another one is a B DNA, right? Sorry, we make a mistake because our DNA is B DNA. So that means this values R is for the B DNA, not for A, because our DNA is not a DNA. Clear? So we have a 34 angstrom here, right? We have a 10 base pair. Okay, I think we have another figure here. Yes, it will be more advanced. So they gives 3 type of DNA. We will add them, right? We will add 2 more. Yes. So B DNA is what? B DNA composed 34. The whole pitch is about the 34 angstrom they gives in the nanometers. That's why it is different. We have a 10 base pair. Okay. A type DNA has the length about the 33 angstrom, but it has what? It has a 11 base pair. The Z DNA having 45.45 angstrom pitch, but it has a 12 base pair under that. Remaining are the C and D. So let's see. C is the DNA. The C-type DNA compose what? The C-type of DNA compose the 9.33 base pair. Okay. And the average length is about 28 angstrom. Okay, that is 28 angstrom. Where on the other hand, the D-type DNA compose 8 base pair. Okay, in the one pitch, and it has a length about 24.24 angstrom. So this is the discussion. Now, but very important thing that we have to know, we have to discuss is, we classify them into A, B, C, D and Z DNA. These four are the DNAs of eukaryotes. This type of DNAs are found in the eukaryotics, and under which, see, our DNA are always rotating, but it rotates like this. Means they are in the right hand direction. Okay, they go in the right hand direction in the clockwise manner, right? So they are the right-handed DNA. Okay, so these are the right-handed DNA. Okay, but the Z DNA is most important. If we discuss about all 5 types of DNA, 2 DNAs are important. One is B DNA and second one is Z DNA. Why? Because B DNA is of our DNA, it's a human DNA, and on the basis of rotation, it is Z. Why? Because all A, B, C and D, they are same as like a right-handed. But what about Z? The Z DNA or Z DNA, that are the left-handed. Okay, that are the left-handed DNA. Clear? So this was the talk related with this session. So now we finish everything. We finish everything, all these 4 topics as I was commit to. So we have finished them. So please, now we have a 16-minute, maximum 16-minute, right, before ending this session. If you have a doubt, as I tell you, that if you have a doubt, put and bombard now them in the comment section. I will give answer of each of your doubt. Yes. Please write your doubt in the comment section. Okay. Thank you, Deep. Thank you. Welcome. Yes, 12 students be ready for 6th chapter. Obviously. Types of DNA. See, we have 2 types of DNA, right? GC-DNA and AT-DNA. If we have a GC or guanine and cytosine more, then it is a GC type DNA. If we have the adenine thymine more, then it is a AT-DNA, right? If we have the structure like what you can say, sorry. If we have this kind of formation, you can divide them according to their structure. If they are right-handedly rotated, there should be A, B, C and D. If they are left-handedly rotated, they are the Z DNA. Again, it is all depend on the length of base pair as well as the diameters and the pitch length. Thank you so much, Atif. Thank you so much. No, I'm not tired. I can keep on, Tirth. मैं नहीं थका. I'm not tired, right? I can move on for and speak for whole night. Yes, size and base pair. Okay, Satya. You can write down the size and base pair. One important thing is this is the yes, this is right information. Okay, no problem. Is there any doubt? Okay. So I make a count 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. It's okay, very clear, right? So this was a beautiful session we done now, right? And it may be possible that every Sunday, Saturday evening, when you have a Sunday for a free, you take this Saturday evening for movies, denote for the NEET preparation, maybe you get more and more marks. So it is very careful and dedicated session for you people, and it might be for so many of time. Okay, honey, you not waste them. Okay, okay. Right, so we are ending this session here. Today I will not tell you that please like, share or subscribe this channel, because if you get something more than whatever other YouTubers says, other teachers says, then like, share and subscribe. Okay, so Jai Hind.